Welcome back again, and in this video we will be talking about the depth structure of a chromosome. First of all, we will be studying the basic structure. After that, we will be going into the schematic presentation of your chromosome. Now take a look. This is the basic structure. Before starting it, you should know the meaning of chromosome. What they help in genetic processes. Now chromosome, if you say, chromosome is the biggest unit of heredity. Okay, it helps for transferring the genetic characteristic from one generation to another generation. You can call it is the most complex or largest unit of your genetics or heredity. The smallest unit, chromosome itself, have DNA is made up of many genes. Now as you can say, gene is the smallest unit of genetics or heredity, while chromosome is the biggest unit of heredity. This is the simplest that. Okay, don't need to confuse in it. Okay, you should know gene will be creating DNA, and DNA will be creating chromosome. Now it is the basic structure. As you can see, the outer thin layer of protein will be called as pellicle. Okay, but we can call it is pellicle. Okay, now pellicle have a liquid or fluids type structure, that is your matrix. In matrix, you can see there will be mineral, water, and enzymes. Okay, this all have matrix. Okay, now as you can see, this is the part, middle part, that we can call it is a centromere. Centromere will help to attach the chromatid part. This you can say arm of chromosome will be called as chromatid, and chromatid will be jointing via centromere. Okay, now if you come into the depth of a chromosome, then this is the thread like structure you can see, and I have diagrammed it as chromoneme, yeah, or you can call it chromonema. Okay, thread like structure inside of the chromosome will be called as chromonema, and a bead like structure will be found on the chromonema will be called as chromomere. It is a bead like structure. Okay, will be called as chromomere. Now via chromomere they will be jointing, and if you want to study the good structure, a very basic look or a depth structure of a chromosome, then you should study during metaphase of cell division. Because metaphase, a chromosome will be having two chromatid. Okay, one chromatid this, second chromatid this. If you come into the division of anaphase, okay, it can be meiotic division or it can be meiosis division. In anaphase time, there will be only one chromatid of chromosome. Then it is obviously understandable that if you want to study the basic or a good structure of the chromosome, then you should uh, study during metaphase division. Okay, now if you come into the depth of a centromere, then you can see the basic disc like structure. It can be called as kinetochore, and kinetochore help during the cell division. It attaches the spindle fiber during cell division. Now take a look. It is the basic presentation. Now is come to the depth structure. This is the schematic presentation of a chromosome. As you can see, this is your chromatid part. This is your chromosome. Sorry, this is your chromosome. Whole schematic presentation of chromosome. As you can see, this is the primary construction. Okay, first one centromere will be attaching the chromosome into the thin part, and we can call is primary construction. Okay, in primary construction you can see the dish like structure will be called as kinetochore, and they help the chromosome in cell division, because they will be holding spindle fiber from pole to the middle stage, middle line. Okay, now this is the outer thin layer of protein, can be called as pellicle, and this is your matrix. As you can see, this is the matrix, and it will have water, enzyme, and minerals in it. Okay. Okay, beside the primary constriction, chromosome also have second one constriction. We can call it is as secondary constriction. If your secondary constriction have nucleolus, then we can call it NOR, nuclear organizer region. Okay, in human, your chromosome number 13, 14, 15, 21, 22, they have NOR region on them. If you talk about what is telomeric DNA or telomere, then chromosome have also the polarity condition, polarity in itself, and the last segment region will be called as telomere. Okay, and the DNA found in it will be called as telomeric DNA, and it's also have in itself telomerase enzyme that is aging belong to aging. Okay, now we have studied that. Now come to the part of your what is the satellite and set. Okay, beside the secondary construction part, the left part of chromosome will be called as satellite chromosome. And if your chromosome have this region, satellite region, then we can call it set chromosome. Set means sign, acid, thymo, thymo, nuclinico region. Okay, so this is the schematic presentation of your DNA, and this I will repeat it in 2 minutes. Okay, this is your primary construction, and beside primary construction, chromosome can also have secondary construction, and the left part beside the secondary construction will be called as satellite. If your chromosome has satellite region, we can call it set chromosome. It means sign, acid, thymonucleinico region. Okay, the chromosome also have polarity in itself, and the last polar region of your chromosome will be called as telomere. And telomere also have a basic characteristics, because it's have sorry it's have guanine base pair mostly, guanine base pair mostly. Then you can recognize the telomere region, and its function to attaching the second chromosome here. Okay, it will help to this chromosome to attaching on another chromosome. Okay, this the DNA founding in this region will be called as telomeric DNA, and in this area also have telomerase enzyme that is belonging to the aging process. This is the basic no depth structure of your chromosome. If you wanna take a look at the basic structure, then this is the basic structure. I think I already told the basic structure and also the schematic presentation. Now you don't have doubts. If you also have doubt, then you can ask me in comments. We'll be coming talking about another videos in my next videos. Thanks.